Right. So um, now we're going to talk about point spread function. We spoke already about point spread function in the optics side. What is the point spread function? How is it formed? And then now we're going to talk about how it relates to resolution. So the point spread function is abbreviated PSF, and I have to apologize in advance because unfortunately Nikon has decided to call their hardware autofocus PFS. So I mix them up all the time. But anyway, PSF is the point spread function, and this is a reminder of what it is. Each fluorophore in our sample acts like a tiny light source. And the light that is emitted by the sample diffracts at the inner edge of the objective. And the inner edge of the objective, therefore, acts like an aperture, which is an iris outside of the image plane. So if you remember, it means that it affects the whole image. So we have irises in the light path. If they are at a place where we have an image plane, then when we close the iris, we can see like a black curtain. But if the iris is outside of the image plane, which is what happens in the case of the inner edge of the objective, then it affects the whole image when it's closed or open. Of course, clearly inside an objective, we're not saying that we can close and open the objective, but the objective has a certain size. So you can understand that the image is going to be brighter or dimmer depending on the size of the inner edge of the objective. So what happens when we have this diffraction at the inner edge? What happens is that there is a 3D interference pattern that is created at the and related to the final image plane, which is where we put our sensor to image. And that 3D interference pattern is complex. So here is our little fluorophore, which is some sort of a sphere, very tiny. And then when the image that is formed in the microscope and the image that we're actually acquiring where we put our sensor, where the image we're recording, in XY, the spots of light is much bigger than the small sphere, and it has all these rings around. And if we do a Z-stack through the small sphere and we look at the Z-stack from the side, this is what we see. We see a rugby man, right? So we see a big bright center that is elongated and all sorts of rings, that complex rings around. So here is the rugby guy, and I played a little bit with AI, and that was already a couple of years ago. Sorry, I have sunlight in my face. So it was a couple of years ago, so it's still some sort of weird stuff. I'm sure AI can do much better nowadays. I actually tried yesterday evening to get a better AI, and I could not get one that was as nice as this one. So I stick to that one. A rugby man that plays hula hoop with lots of rings around them. So the problem that we have here is that this is what we get when we have one fluorophore. But what happens when we get more than one fluorophore? And clearly in our samples, we have more than one fluorophores, right? What happens to all these rings? And what happens to this big bright center here? So this is what we're going to look at now. How does this point spread function, the blob with the rings, relate to resolution? So you can see here, this is the bright center of the point spread function. If we would push the brightness and contrast, we would see the rings. The rings are so much dimmer that it's a little bit hard to see them. So here is the bright blob that is in the center, the rugby ball. And you can see we have two fluorophores in our sample. So we get in what we record on our sensor is two blobs. And now they are a little bit far away from each other because in the sample, the two fluorophores are quite far away from each other. But now, if the two fluorophores somehow get closer, you can see that the main blob of the point spread function, they start touching each other. And now two fluorophores are at a distance where we have actually one diameter of point spread function. And so the point spread functions are not touching. And this corresponds to one diameter of one point spread function. And so in here, we are still resolving the blobs. But if we keep on going and it gets closer and closer, we stop resolving. So clearly the size of the point spread function is related to what we can resolve or not. So this is the distance between the two dots in our sample is actually the size of the point spread function. We can still resolve it. But if we have now half a point spread function diameter, so one radius, so just half this distance here, if the two dots in our sample are at one radius of the point spread function distance, then we cannot resolve them anymore. And if clearly if they are closer, we just see one dot and we cannot resolve. So the resolution in the macroscope is the smallest distance between two points that can be reliably distinguished. And the smallest, when we have a small distance, we have a high resolution. So when we can put two, it's a little bit counterintuitive, but a small distance is a high resolution. So when we have two dots in the sample, they can be very close to each other and we can still resolve them. It means we have a small distance between them, but we have a high resolution. Okay. So all objects that are smaller than the point spread function appear in our image to have the same size of the point spread function. So we cannot make because this is the image that we form from one little dot, it is not possible to have smaller dots than the point spread function because the point spread function is what the image that the microscope makes of a tiny dot. So even if the dot is tinier, it's anyway going to make, this is the limit of our resolution if the size of the point spread function. The smallest distance that can be resolved is the radius, so half the diameter of the point spread function. And you can see in here that this is what we are calculating with the equation that I gave you. So here is our little dot in our sample, it fluoresces, and then this is the image that we're getting, the 3D image that we're getting and that we're measuring with our sensor. This is in the XY plane, and this is if we do a Z stack through our little bead and we look from the side, this is what we're getting. And if we draw a line in here, this is the curve that we get. So we see the bright center in here, and then we can see here the rings around the bright blob. So if the two dots in our sample, the two fluorophores in our sample are at a distance that is more than one radius of point spread function, we can resolve them. If they are at one point spread function, one radius, we can still resolve them. But if they are closer, we cannot resolve them. Okay, so here is this is the diameter of the main blob. And here is the radius of the main blob. So this is the optical resolution. And when we use this equation, 0.61 lambda divided by NA, this is what we calculate. All right. So I don't know how this 0.61 came about, but this is part of the equation. And lambda, as I said before, in this course, we're going to use the wavelength of the emission of the fluorophore, okay, for everything. And this is, again, to make it simpler. And then the NA is the numerical aperture of the objective. Now you know that. So this is this distance, basically. It's the radius of the main blob. So they are, again, this equation is called the Rayleigh equation. It was found empirically. So he was eyeballing, basically, and going, oh, now I can see, now I cannot see. Clearly, it was depending on his eyes and things like this. Maybe the sunshine outside, I'm not sure. But there are many different equations. As I said, the full width half max is the FWHM is often used to calculate the resolution. What does that mean, full width half max? This is the maximum intensity of the main blob. And in here, we have half of the main intensity. And here we have the full width at half maximum. This is what it means, full width half max. And so this one, the calculation for this, if you would want to calculate this distance, is 0.51 lambda divided by NA instead of 0.61. So clearly that one, the full width half max, is more optimistic because your blob will appear to be smaller because you're calculating here instead of calculating here. It's a little bit smaller. So it's more optimistic. And now we have another equation. The Abbe equation is half of the wavelength divided by the numerical aperture. And that one is theoretical. So it's sort of the limit of, it's a very limit, but it's really a calculation. And then we have Are we going to use the excitation wavelength? Are we going to use the emission wavelength? In theory, if you use a camera-based system, you should use, no, sorry, if you use a wide field system, you should use the emission wavelength. If you use a confocal, whether it is a single point confocal or a multi-point confocal, you should use the excitation wavelength, okay? And that is because the excitation is also focused. So that is what we should use normally, but in this course, we're going to simplify things. And the Z now is going to be also the radius of the main blob, but in the Z direction. And the equation is the one I gave you. So 2 lambda multiplied by the refractive index of the objective and divided by the square of the numerical aperture of the objective. And you know that this is the depth of field. And again, this equation is valid for wide field. So it is this distance. But if you want to use a confocal and your pinhole is bigger than 1 area unit, this is the real equation. And again, in this important equation for light microscopy page that I have on Canvas, there's a lot of different references if you want to know, including a very nice PDF by Zeiss that this is coming from. So you can have a look at that if you want, but you can see that it is a complex equation, much more complex than the wide field equation. But in this course, to simplify things, because I don't want to confuse you, we're going to be using this equation for everything for Z and this equation for everything in XY. So we're going to calculate the radius of the emission point spread function for everything, for all systems and for X, Y, and Z. Okay, so this is the radius and this is the radius, and these are the equations that we're going to use in this course. And the pinhole, when we close the pinhole, if we stay at more than 1 area unit, what we're doing is we're not including, we're not calculating sorry, we're not increasing the resolution, we're increasing the contrast. You can see in this, which is the correct equation for pinhole open more than 1 area unit, there is no size for the pinhole. So the pinhole, closing the pinhole has no effect on the XY resolution if the pinhole is open at more than 1 area unit. But it has an effect on the contrast, which is extremely useful, actually. You can see if you have a low contrast, you can have excellent resolution, but you can actually not use it. So the contrast is clearly very important in microscopy, and we will talk about this later. So, so what this 1 area unit, what is that? When we use a confocal, we have a pinhole. And so when we have a pinhole, we often talk about area unit, and it's often 1 area unit. So why 1 and why area unit? So I remind you for a single point confocal, but the principle is the same for multiple confocal. We have 1 spot of excitation. We have a focused excitation. We have a focused emission. The excitation light goes through the scanner, through the objective to the sample. This little spot of light is excited. And then the emission comes back through the objective, through the scanner with the dichroic mirror. Now you're a super expert in all these things, right? And the emission goes to the pinhole and then goes to whichever detection system you have, which could be a filter-based or a spectral detector. And so what happens in here is that we're blocking all the light that comes out of focus. So if you remember, this pinhole stuff is actually placed at an image plane. So this is a field stop, okay? So if the pinhole is more open, more closed, what happens is that it acts as a curtain around the point spread function, because this is what we have. This is an image or a sample of our tiny spot here. So it is the point spread function, has the point spread function shape. So we put this pinhole and we remove part of the pinhole. So you know that we have this complex 3D pattern, interference pattern that is formed, and you know now the shape that this has. So the image that is here has this shape. And what we do when we have a pinhole, is that we basically remove all the rings, okay? And when we remove all the rings, what happens is that if something else is, another fluorophore is a bit out of focus nearby, the main blob is going to be blocked out. So most of the light is in the main blob, and so most of the light is going to be blocked out, and this is the way we reject, this is the way the pinhole rejects the out of focus light in our microscopes, in our confocals. So we have only we're left with only the main blob. But you can imagine that if we close the pinhole more, we're going to lose a lot of light. If we open the pinhole too much, then we're getting the rings in, and we're getting also the out of focus light, the light that comes from the main blobs from other fluorophores. We get that in as well. So we're decreasing our contrast. So the size of the pinhole matters in how much contrast we have. And this is what do we want, which is the best size. Um, you remember that the size of this point spread function depends on the wavelength. So it's 0.61 lambda divided by NA. The lambda is the wavelength of the light. So if you image a blue fluorophore or you image a far-red fluorophore, the point spread function is going to have a different size. The point spread function of a fluorophore with a longer wavelength, like for example a far-red fluorophore, will be bigger than those with the blue fluorophore. That's why I said that the blue fluorophores, they deliver better resolution because the point spread function is smaller. And the way people have managed to talk about the ideal pinhole size, we will discuss that a little bit later, the ideal pinhole size for a confocal, to standardize the size, because it depends on the wavelength, to standardize it, we basically are calling this a 1 Airy unit. So 1 Airy unit, the Airy unit is basically the size of the pinhole, how open or closed it is. But 1 Airy unit just means from the first dark ring here to this the diameter of the first dark ring when we look at these complex rings. So the diameter of the main blob is 1 Airy unit, regardless of the wavelength. Okay, so this is what 1 Airy unit means. It means you're rejecting the rings, but you're keeping the main blob. If you close more, if you have a pinhole that is at 2 Airy units, you're going to get some of the rings and decrease the contrast. If you have a pinhole that is smaller than 1 Airy unit, you're going to lose a lot of the light because you are shutting off part of the main blob. This is what 1 Airy unit means. So we will discuss a bit later why we want to, when do we want 1 Airy unit, when do we want more, or when do we want less. It is not the case at all that we set 1 Airy unit for everything. We make decisions. Yes. So 1 Airy unit is used to normalize the physical size of the pinhole, and the physical size of the pinhole, because the objective has magnified the light, the physical size of the pinhole at 1 Airy unit is this, which is the radius of the main blob, multiplied by 2, because now we want the diameter. We don't want just half of it. So the resolution is this. Now we want the whole diameter of the blob, so multiplied by 2, and multiplied by the magnification of the objective, an extra lens if you have. And so the whole thing is in microns. So sometimes when you look at your confocal and you see the 1 Airy unit, you can change this up and down. You will see there the size, the physical size of the pinhole, and that is in microns, and that's because it's multiplied by the magnification. Okay. So now we know what 1 Airy unit means for confocal users, and clearly this is true both for multi-point and single-point confocals. So the resolution, Robin.
Yes, super quick question. Maybe I
Yes.
should ask this to the one that has the microscope. But since the area unit is different in terms of micron for every wavelength, if you have a confocal system like the one I'm using, where you set, for example, area unit 1 and then image 3 different fluorophores, will the pinhole change all the time? Or is it because sometimes you see that it's standardized to 488, for example? Is the typical thing that it changes if you do it sequentially, or have you just set one pinhole that is the same for all the image acquisition?
Yeah, that's an excellent question. And different microscope manufacturers have made different decisions for this. So it used to be the case that you would have different pinholes you would have one pinhole per detector. But this was abandoned a long time ago because it was making it very difficult to compare the different images. So now there is one pinhole on a single point confocal. There is one pinhole, one size of pinhole. On a multi-point confocal, we have also one size of pinhole. We have thousands of pinholes, but they're all the same size. So we have one size of pinhole per image. And there, the manufacturer has to decide, am I going to calculate the pinhole size when you click on one area unit? Is this going to apply to, let's say, if I have three different fluorophores, is it going to apply to the short wavelength or the long wavelength or the middle wavelength? Yeah. So there is a default for each manufacturing company, and you can change it. It's sometimes a little bit hidden. Zeiss, and I'm not sure about Leica, but Zeiss sets the pinhole for the shortest wavelength, which means that the long wavelengths are going to be dimmer that if they are at one area unit. So you can say the long wavelength, for the long wavelengths, the pinhole is going to be at lower than one area unit, but the short wavelengths with the one area unit. Nikon has chosen the other way around. So for Nikon, the long wavelength is the one that applies. So they're going to set the one area unit for the long wavelength, and then maybe the short wavelengths will be have a bigger pinhole that they should have compared to one area unit. So you will get more light in, but a little bit less contrast. Is the way it is. But you can change these things. You could set, if you image sequentially, you could change this, but it's not recommended because it's then harder, let's say, to compare the images since they all have different pinholes.
Okay, thank you.
You're welcome. Yes. Where is my mouse? It's gone here. Okay, so how does this thing with the point spread function and the resolution affect the sample design? And why do we want to do this reverse thinking thing? So you imagine that you have you have a sample where you have quite a lot of small dots and they are very crowded. And so they're close to each other. You really need to have a high resolution to be able to resolve these objects, right? Now, if you have the same object, so they are small dots, but they're far away from each other, you can make do with low resolution because they are far away from each other. And what matters is the distance between the objects. It's not the size of the objects. If now you have big objects, they're very close to each other. For example, nuclei in skin, in the upper layer of skin, they're very close to each other. Everything is packed. If you want to count the nuclei, for example, and you label them with DAPI, you need a really high resolution needed because what matters here is the distance between the nuclei, the two adjacent borders of two nuclei. So you can use a trick here and say, instead of labeling with DAPI, I'm going to use something that labels the nuclear membrane, and I'm going to have this big empty space in between. And so now what you need to count your nuclei, what you need to be able to resolve is really the distance between one side of the nucleus to the next side of the nucleus, which is much larger than the distance between the nuclei. And this way you can get away by changing your sample preparation and the way you label the nuclei, you can get away with using a lower resolution and maybe enjoy a larger field of view. Okay, because now it's the distance here that matters. So you can see how, based on which resolution you need, you have choices to make, basically, and that can influence the way you image your design your sample. So the required resolution is not related to the size of the object, but to the distance between the objects. Now, if what you want to do is to measure the size of an object, then the distance between the objects, the objects is the one side to another. So sometimes the required resolution is the size of the object. This is when that's what you want to measure. If you want to measure the size of the object, what you need to resolve is one side of the object compared to the next side, the other side of the object. So, but if you want to count objects, it's the distance between the objects that matters. So the design of the sample influences the resolution that is required to answer the scientific question. So we have a crosstalk there between scientific question and the design of the sample. So you do reverse think your experiment and take into account your knowledge of the biology of the sample so that you make decisions, so that you not decide that you're going to be counting tiny, tiny objects that are very crowded. If you know that the microscope is not, it's not possible for the microscope to do it, then you change your scientific question. But maybe you can also adapt your sample design around your scientific question. So, okay, so now I'm going to, let me see. Yes, okay. I'm a tiny bit behind now. That's right. Here is the conclusion for this little part. We have, so the resolution is the smaller distance that can be reliably measured. And again, this depends on how we analyze the image. So please analyze your images before acquiring tons of data. And without super resolution, it is not possible to resolve distances that are smaller than the PSFS radius, so half of the diameter. And we'll talk about super resolution later. And the objects that are smaller than the point spread function appear to have the size of the point spread function. So it's not possible for the microscope to measure objects that are smaller than the point spread function. They all look like they have the size of the point spread function. In this course, we will use the same equations for camera-based and single-point confocals for the XY optical resolution. So I remind you, optical resolution means the resolution that the objective is giving us. And we'll see later why this is important. So this is the 0.61 emission lambda divided by NA, the numerical aperture of the objective. This is for the XY optical resolution. And the Z optical resolution is the depth of field. So 2 emission lambda multiplied by the refractive index of the objective immersion medium and divided by NA square. So we use this for everything. But I want to remind you that when you close the pinhole to 1 area unit on a confocal, you do not increase the resolution. What you're increasing is the contrast. Things are different when we have a pinhole close smaller than 1 area unit. But for up to 1 area unit, you do not increase the resolution. Okay, so now what we're going to do is that we're going to go to the workshop Zoom. And we will have some little quizzes and discussions and things like this. And then we will come back here for another lecture at 1.